Good afternoon, everyone. I'm Prarthini, and this is my honours thesis. I'll be talking to you today about the conservation status of the Malayan horned frog, Megophrys nasuta in Singapore. Now, I know I'm the last speaker, and you're probably a bit tired of hearing all these talks, so I thought I'll start with this. Is anyone able to spot the frog in this picture? You have 5 seconds. 5, 4, 3, 2, 1. You find it? So this is indeed a unique and exquisite frog species, just as unique and exquisite as the many amphibians that we have in the world today. Unfortunately, amphibians today are facing a crisis. Many species are going extinct at unprecedented rates. So here is a reflection of the amount of amphibian species which are critically endangered or are facing extinction. With that, massive work is being undertaken in order to discover more about their life histories, to detail their conservation statuses, to find out what are the habitat requirements which are necessary for their survival in the wild. Now, in Singapore, we have about 30 species of amphibians, and not very much information is known about the kind of requirements necessary for them to stay alive in the wild. And although we do have information about their conservation statuses, this information is often not up to date. An example of such a species is the Malayan horned frog, Megophrys nasuta. So this is a pretty large frog. The females can go up to 16 cm. It's got prominent triangular projections, and it's generally brown in color. And these morphological characteristics, the triangular projections, the markings on its back, the fact that it's brown and it looks like a leaf, actually aids in its camouflage abilities. So if you're in a forest floor and you look on the ground, you probably won't be able to see the frog unless you're looking hard and closely. As for its young, we know from literature that the eggs are large, they are white. We know that the larvae they're distinctively elongate, and a unique thing about this species, which is unlike any other species in Singapore, is that it's got a very unique mouth. So the labia of the tadpole is enlarged, and that aids in its feeding strategy. And we also know that the larvae take about 2 to 2.5 months to reach metamorphosis. Now in Singapore, we are aware of the fact that the Megophrys nasuta is distributed across the Central Catchment Nature Reserve and Bukit Timah Nature Reserve. So although we have this kind of information, again, current up-to-date information about its conservation status, as well as the kind of habitat requirements necessary for its survival is not known. Now with that in mind, this study was conceived. I had 2 main research objectives. The first was, what is the current conservation status and the distribution of Megophrys in Singapore? And the second was, what are the habitat requirements necessary for the survival of extant populations in Singapore? How did I do this? Now I first had to get my hands on all the historical records of Megophrys, and I had 2 main sources of information. The first was, in 1993 to 1996, UN Rajathurai conducted a comprehensive survey of all the amphibian species in Singapore. And I got a map from their study, overlaid it on Google Earth, and got the GPS coordinates. From that, I had 12 historical sites. My second source of information was the museum. So I contacted the museum curator, got all his records, and that gave me an extra site. Altogether, I had 13 sites. And to those 13, I added 6 streams which are basically in the vicinity of these historical locations. So these are my 19 sites, spread out across Central Catchment as well as Bukit Timah Nature Reserve. I used a combination of auditory as well as visual encounter surveys, and I established transects at each of these sites, and each transect was then surveyed 4 times throughout my thesis period. I also sought to look at the population size of Megophrys in Singapore. Now, unfortunately, in one of my sites, only in Fern Valley did I regularly encounter individuals. So it was the only place in which I was able to do a population size estimate. In Fern Valley, I did a modified mark recapture method, which is basically looking for the frog and determining whether I've seen it before. Because each individual frog can be distinguished by the markings on its back, the presence of the tubercles, which are these black dots that you saw. So some of the data that was used in my thesis was obtained from my previous study in my 3rd year. Now, following all those surveys, I conducted habitat characterization of all my survey sites. So I went out 3 times to get all these measurements, and these are pretty standard measurements taken for habitat characterization. And finally, in data analysis. So in order to find out the distribution of Megophrys in Singapore, I used an online software to determine the extent as well as the area of occurrence. And I used the Schumacher-Eschenmayer estimate for a population size estimate in Fern Valley. And as for the habitat characterization, I used generalized linear mixed models in order to determine the relationship between the environmental parameters as well as the occurrence. Now, after saying all that, what did I actually find out? So altogether, I conducted 300 surveys across 75 different transects. And what I discovered, unfortunately, was that the number of sites in which Megophrys is currently present has decreased. So originally from the Theo and Rajachari study, I had 12 historical sites. Unfortunately, only 3 of those 12 have confirmed presence. So the site provided by the museum, Leisure Valley, still had confirmed presence, while I discovered a new population in Bukit Timah Nature Reserve at the catchment path. From these current locations, I then looked at the extent of occurrence, and I did a historical comparison with the data from Theo and Rajachari study. So what you see here in the light pink polygon, or light gold polygon, or whatever, is the past extent of occurrence, and the dark purple polygon is the current extent of occurrence, and you can see that it's shrunk by about 50%. This is the same for the area of occurrence. So on your left is the past area of occurrence, and on the right is the current area of occurrence, and that too has shrunk by about half. The Schumacher-Eschenmayer estimate gave me a possible population size of about 63 frogs, with my 95% confidence limits running from 35 to 252 individuals. Now, why is this confidence interval so huge? This is due to the low number of recaptures that I had. So I counted about 23 individuals altogether in Fern Valley, and I only had 4 recaptures, which is likely because of detectability, and that would have inflated my population size. These are the results of the GLMM analysis. Now what you see here, these dots, those are the effect sizes, and that basically tells you the effect or the impact of the relationship of the different environmental parameter to the occurrence, your response variable. Your error bars that you see are the 95% confidence intervals, and what that basically means is that how sure are you 95% of the time that this relationship is actually true? And what that showed me is that dissolved oxygen and bank vegetation seem to have a significantly positive effect on occurrence, which is reflected by the fact that they didn't overlap 0. Now, analysis of my raw data also showed me that refuges accounted for 17% of the variation in my occurrence data. What that means in English is that essentially every time I encountered a frog, there was also a refuge in that transect. So this showed me that these 3 variables, these 3 variables, dissolved oxygen, bank vegetation, and refuges, were important in explaining the occurrence of the frog. Now to explore these relationships further, I constructed GLM analysis plots. What we see here is how the relationships differ across the 19 different sites. So you have your response variable, and my explanatory variables. And on average, what we see is that an increase in dissolved oxygen or an increase in bank vegetation results in an increased likelihood of Megophrys nasuta presence at that site. That's a lot of information to take in. So let's pause and recap. What were the objectives of my study? The first was to determine its conservation status, and the second was to find out the habitat requirements necessary for its survival in Singapore. What did I find out? That it's likely possible and needful for the conservation status to be updated from endangered to critically endangered. Why? This is based on the evidence of decline. The decline in the number of sites where it is currently present, the decline in the extent as well as the area of occurrence, and likely the decline in population size as well. All of these declines point to an overall decline of Megophrys nasuta in Singapore, as well as increased fragmentation between populations. As for the habitat requirements, I saw that dissolved oxygen, bank vegetation, as well as refuges seem to be important in explaining the occurrence of frog at a certain site. Why is that so? So I'm postulating that dissolved oxygen is important for the tadpoles, because one of the things that I realized when I was contacting frog breeders is that they told me water quality is actually more important for the tadpoles rather than whether it gets to eat or not. As for bank vegetation, in general, for most frogs, it acts as a calling site or a resting site, and that is no different for Megophrys. Finally, refuges in the stream serve the same purpose that bank vegetation does as well. Now, of course, like any scientific study, there are some caveats and limitations, and I did talk about one of it briefly. So the first was that I had a low recapture rate for our population size, and this, mind you, is the first time that anyone has ever done a quantitative abundance study on frogs in Singapore. So although I wasn't able to detect the frogs, and although my population size would have been inflated, this information is still useful because it gives you a sense of how many frogs there might actually be in one particular place. And of course, like almost any statistical analysis, greater sample size would have afforded me greater certainty with the results that I got from my habitat characteristics. In conclusion, the conservation status of Megophrys nasuta in Singapore should likely be updated from endangered to critically endangered, and this of course is based on the evidence of decline. Next, dissolved oxygen, bank vegetation, and refuges appear to be important in explaining the likelihood of Megophrys nasuta occurrence at a certain site. There are some prospects for future study of Megophrys in Singapore. So the first, of course, would be to properly get the population size of Megophrys in Singapore and to be able to delineate carrying capacities of each present site. Now, during my surveys, I was fortunate enough to encounter a pair of amplexing Megophrys, and that led me to think about what is the kind of reproductive ecology of this frog, because it's not very much studied either here or in the rest of the world. And finally, also during my studies, I encountered a number of individuals which had very strange lesions and growths on their nostrils as well as their backs. And that led me to think about what are the kind of threats that these populations face and the need to properly identify them. Of course, all this information, like habitat information, will be necessary in order if we want to conserve this species and protect the species in Singapore. That's all. Thank you.